Okay, Zach, so we will record it, but if you have anything against something, so some people showed and published data and didn't want it to put it in the internet, then we can do it both ways. So we can not put the presentation, or we can cut a piece of it out of the presentation, editing it, if you have something that you don't want to put.
No, it should be fine.
Okay?
Yep.
So, okay. Great. Yeah. So I will make a short introduction for the people that eventually don't know you. So as is my case, so I know your work, but not we never met.
Yeah.
So it's a pleasure and an honor to have today Zach Adelman. So Zach made his PhD in Colorado. Who was your advisor, your mentor by that?
So my co-advisors were Carol Blair and Ken Olson.
Oh, okay. So great. With that group, so both Carol and Ken were really the pioneer people in the interaction, who really created the field of interaction between mosquitoes and dengue virus. Yeah. So most of seminal work were done by that group. So he has had really, he came really from a key place in vector biology, globally speaking. So, and after completing his PhD, I think he worked with Tony James, no?
Yes.
Yeah, Tony. So we have someone, Vanessa, that's now in Tony James, that's in the audience. So she worked with Gabriela and is now in Tony's lab, and Margrethe also. So Tony is known from several people, which is a lab that had also key contributions in transformation of the mosquito, including the first. This was a kind of key moment for the whole career of Zach, that is has a guiding thing, that is a profound knowledge and a very deep expertise in mosquito transformation. So he's possibly the gold medal in the number of Aedes mutants, or something like that. Maybe the silver. I don't know the count exactly, but it's one of the top guys in the there. Hi, Marcelo.
Hi. Good seeing you.
Yeah, I already started the presentation. So and from Tony James, he went to Virginia Tech, where he was following that trail of working with mosquito transgenics, but also using it as a tool, both with the long-term idea of control mechanism, but also as a tool to study very relevant points of mosquito biology, such as interaction with virus and male sex determination, and also, at least for me, it's very interesting, heme and iron and metal transport, that where he made very interesting contributions using powerful molecular screens and genetic transformation. So in fact, he has also a very important contribution in the discussion and the formulation of regulations for the research and eventual implementation of transport tools using mosquito control tools using genetic transformations, and so that's a sensitive issue, and he's one of the guys that is I think making the point in that area. So I think that is so. I'm not going to use much more of the time, and we can start. So Zach, thank you very much for accepting the invitation. It's a pleasure to have you here. And I hope we'll have you here physically someday in not a long future.
That would be very enjoyable. Thank you. Well, thank you so much for the kind introduction. That was really too generous. And thank you everybody for taking the time out of your days to come in and listen to the presentation I'm going to give today. It's really just really great to meet so many people that I've followed their work, like Pedro, that for so long, and to people that I haven't seen in a long time, like Luciano. It's really great to just to have everybody together like this. So I'm really looking forward to, as I go through the slides, if something catches your eye and you have a question, just ask, interrupt, very informal, it's perfectly fine. I want to have conversations with everybody today, so just interrupt when you see something interesting to you. And so with that, I will get started, and we'll see. So hopefully everybody can see the slides, everybody can nod or thumbs up. Okay, so the work that's done in my lab, I won't be able to go into every project that we've got. I don't have enough time for that, but I will tell you a few of the projects. But this is in general the focus of my lab, if you can call it focus. I call it the lack of focus of my lab, but this is just the way it is. As Pedro mentioned, we are working with Jake 2 on sex determination in mosquitoes and how to engineer sex factors. I'm going to present some of the work that we're doing on mosquito physiology, because I know that's of special interest to several of the researchers in this group, and in terms of heme and iron detoxification. We're doing some work in immunity in collaboration with Kevin Miles, looking at different genes involved in RNA-based silencing to dengue viruses and other viruses. Also some work that we've done with Eric Calvo's group at NIH, and also some with Margaret looking at parasite host factors in the mosquito. I won't talk very much at all today about some of the projects we're doing on DNA repair and engineering the genome and gene drive, but we will talk a little bit about our genetic disruption of female flight. And as Peter alluded to, all of these projects are kind of linked together in the tools that we use, which is CRISPR-Cas9 in traditional transgenesis, where we can manipulate the genome and remove segments or add segments as well. And so we've really got these genetic tools to the point where we've always dreamed of back in 20, 25 years ago, when we thought that if we could just do all of these things, we could answer so many questions. And it's really great to be able to start doing some of them with these tools. So I don't have to go over these slides in much detail. You guys are very familiar with dengue and where it is and its toll. So I think we can just skip right over why dengue is a problem, and this is why we study Aedes aegypti, because of dengue. It does transmit chikungunya and yellow fever and Zika, but really we study it because of dengue, because that is the virus and group of viruses that are continuing to cause the most trouble in the world, as you all well know. And so again, to reiterate, here's our life cycle of our mosquitoes. They alternate between taking blood and then hiding in these kind of areas that you might find in your backyard, tires and drain spouts and bird baths and pots and whatnot, and then coming back to find the next person where they might pass on that virus. So we're very interested in basically everything a mosquito does that's part of this process, from acquisition through transmission, trying to understand how we can stop even just one of those events from happening, and you could potentially break the transmission cycle. So we have on the acquisition side, the mosquito has to first acquire blood, and so that requires a lot of different salivary factors. It requires behavioral factors, and we do have some projects involved in manipulating salivary gland biology to try to decipher the roles of different salivary gland proteins in this process. And then once that pathogen makes it to the gut, right, that's a dead end for most pathogens, things like HIV or flu virus or even coronavirus now, COVID-19. If it's struck by a mosquito, is going to be digested. It's going to meet a horrible fate or a pleasant fate, as the case may be. But some viruses like dengue and, of course, parasites like malaria have figured out how to escape the gut and then continue on with their life cycle. Meanwhile, the mosquito has to survive this process of ingesting her whole body weight of blood or more by detoxifying the heme and iron that comes out of that, surviving long enough to lay eggs. Meanwhile, the pathogen has to then escape the gut and then reinfect the salivary glands, whether that be dengue, whether that be malaria parasites, so that on the next blood feed, you can go on to transmit. So again, interrupting any of these biological processes could potentially result in a mosquito that is then unable to transmit those pathogens, either because she doesn't live long enough or because she's lost the ability to support replication. So here's a few hypothetical questions, right? So we imagine that if a mosquito dies after drinking on blood, because if I tried to drink my whole body weight in blood, I'm pretty sure I would drop dead on the spot. And we would like the same thing to happen to our mosquitoes. Then they would not be able to transmit anymore, right? And so one of the questions we had was, how exactly do mosquitoes detoxify these blood meals? They have to separate heme, and all this wonderful work that Pedro has done has shown that they have to actually detoxify the heme in addition to binding a lot of it from the PM. So we have this really nice figure that my graduate students put together as part of a review that they wrote a few years ago. This is from Siobhan Whiten and Heather Eggleston. They both moved on. But we have this kind of model where we have a lot of the heme might be aggregated in the peritrophic matrix. We talk about different arthropods might drink different amounts of heme. And then some of it is modulated in terms of the antioxidant activity by things like XA. But some of that heme, and a good chunk of that heme, has to be taken into the cell, where some of it is required to activate the 20E pathway that will then eventually stimulate oogenesis. And then much of that has got to be degraded, where biliverdin is then sent out to the lumen, releasing a lot of free iron. And that free iron has to be dealt with as well. And of course, there's transferrin-bound iron in the blood, along with heme-bound blood. And that transferrin-bound iron is released, which can then be taken up by the cell as well. So in flies, this iron uptake process needed by the transferrin Malvolio. And in Anopheles ablomanus, ortholog of that, so these are basically the same molecule. And we looked for this really, really hard in Calcine mosquitoes and could not find it. And so we think that Calcine mosquitoes are taking up free iron in a different way. They're not using this transporter here, and we were curious as to what that might be. In the meantime, once it's in the cell, this free iron then has to be loaded into the ER, where it can be packaged in ferritin and then into the hemolymph, where it can be transported to the tissues that require it. Alternatively, free iron itself could be sent out, but we're not sure about that process either in terms of whether transferrin binding the hemolymph or not. And so basically we set out to identify these first steps. How is heme getting into the cell? How is molecular iron getting into the cell as a first step? Because if it didn't get in, then we would basically leave it out here, and the cell that needs it wouldn't get it. We might not stimulate oogenesis. And we might have off itself. We can't load it. So this is our goal, is to try to figure out these missing pieces. And to start, we tried to develop an assay that allowed us to quantify the amount of cytoplasmic iron in a mosquito cell, because quantifying molecular iron itself is difficult to do. And so what we did was we generated a reporter assay harvesting the light chain promoter from the exogypti-ferritin gene, right? So here's the heavy chain gene and a light chain gene. They're arranged just like this in the mosquito genome. They're separated by about 600 base pairs. We took that fragment. We know in the mosquito, this just is induced by iron and by blood feeding. And we connected it to the firefly luciferase gene. And then we transfected that into cells. Control expresses renylluciferase all the time, and measure the ratio of firefly to renylluciferase, right? And so in the absence of any iron stress or iron induction or blood meal, the expression is very low from this promoter. And so you end up with a very high value down here and a very low value up here, which gives you a very low number, which is what you see here. But as we increase the amount of iron, and this is ferric ammonium chloride that we added in at either 20 micromolar, 50 micromolar, or 100, you can see this kind of stepwise increase in the amount of luciferase that we can detect in these cells. Because again, bottom value doesn't change, but the top value gets larger as this luciferase gets induced. And we found that this reporter was not activated to any real extent by copper. So here's that same 100 that we see here. We didn't see any change whatsoever. When we boost it to 1,000, so 10 times higher, we just barely start to see some activation. So we kept all of our assays at this 100 micromolar. So it's basically not responding to copper. It doesn't respond to zinc. It's specific to iron in these cells. And then we tried to look at mechanisms of which types of transport are necessary. So we used some inhibitors of the Clathrin pathway and some other uptake pathways, and saw that both kind of play a role in the uptake. So this has got to be taken up into a vesicle and then taken into the cell through what we presume is some transporter-mediated event. And so what transporter is doing that, right? So we couldn't find N-ramp Malvolio transporter. So our next best guess was to look at this family of zinc iron importers or zinc transporter-related families. And we just basically mined the genome for as many of these as we could find based on their orthologs in flies or humans or yeast. And then basically made double-stranded RNAs to all of those genes, transfected those into cells, and then after a few days transfected in our reporters, and then measured our luciferase levels. And you can see here's GFP double-stranded RNA, so that's our baseline. And most of these genes, when you knock them down, they don't do anything to this ferritin reporter, suggesting that those are not involved. But we did see a few genes that consistently gave us either highly elevated values, suggesting that there is much more iron in the cell, or decreased values, suggesting that the amount of iron had decreased in those cells. And so we're really interested in these three genes here. When I've highlighted the clades, two of them, these two here come from this ZIP family. And I've aligned here at the human Drosophila, Anopheline, and Aedes genes. And you can see here's this ZIP11, which is here in this D. And then here is this ZIP13. Again, it has close orthologs in Drosophila and Anopheles and humans. And it's this 14762 value, whereas this ZNT7 gene, which has close orthologs again in Drosophila, Anopheles, and humans, this ZN7, showed this decreased iron. So we're interested in how these might behave in the mosquito itself rather than in cell culture. And so that's basically as far as we got with this project when we moved from Virginia Tech to Texas A&M. At that point, a new postdoc, Hitoshi Tsujimoto, took over this project. And he started looking at the localization or expression of these three genes in the mosquito. So here we have the midgut, Peyton tubules, ovaries, and then the rest of the body, looking at expression of either ZIP7, ZIP11, or ZIP13. And basically what he saw was that they are expressed to some extent in all of these tissues, although ZIP13.
Sorry, Zach, this is sugar-fed mosquitoes.
This figure here is sugar-fed. I'm going to go to blood feeding on the next panel here. So this is in the sugar feeding state. ZIP13 has the lowest expression, and then there's this basal expression in basically all these tissues. When you induce a blood meal, so here is 0 hours, 6 hours, and 24 hours for either ZIP7, ZIP11, or ZIP13. What we can see is that the expression does change a little bit. For ZIP7 in the midgut, it goes up very slightly, maybe a little bit more in the Malpighian tubules at 6 hours, and then interestingly, by 6 hours, by 24 hours, the expression had reverted. And the ovaries are kind of on a delay where it goes up by 24 hours, but not early. So that was kind of interesting to us as a potential importer that might be recruiting iron to the Malpighian tubules during mid-digestion and recruiting iron to the ovaries later in digestion. And then ZIP11 didn't really show much, except we did see the same type of late induction, this time in the Malpighian tubules, but not much in the ovaries at all, and not much in the midgut. And then we did see some induction in other parts of the body. We don't quite know where that is. It could be fat body, it could be others. We haven't instigated that yet. But we see the strongest induction from this ZIP13, which is barely expressed at all prior to a blood meal, but by 6 and 24 hours in the midgut, the expression increases dramatically. This is more than 40-fold, and then not much in many of the other tissues. Then Hitoshi wanted to know if this was regulated by a blood meal or if it was regulated by iron alone. And so what he did was he generated an artificial meal that contained only ATP and buffer, but also with iron. And what he can see is that this gene, ZIP13, is induced when the only component of the meal is iron, and there's no protein in the meal at all. So this seems to be iron-regulated activity. So then we asked the question for ZIP.
Have you tested it with him also?
So we have struggled a lot with trying to do artificial feeds with heme alone because it's so insoluble that we typically get substantial mortality when we try to just dilute heme and solubilize it in either DMSO or in sodium hydroxide. We haven't been able to do that experiment, and I'd love to hear any suggestions you may have.
Yeah, it's a kind of trick.
Yeah.
That would be boring for the audience. So I will send you an email with some a detailed protocol on that.
Okay, that would just be wonderful because yeah, our students struggled with high levels of mortality when we tried to do those feeds with heme alone at the concentrations we were able to get it solubilized. So that would definitely be very interesting. So what Hitoshi did next was he made double-stranded RNA to the ZIP13 and inject that into mosquitoes and then offer them a blood meal. And so the first two panels are calcine fluorescence. And those of you familiar with calcine or not, so this is a molecule that fluoresces, and then the fluorescence is quenched by the presence of cytoplasmic iron. Okay? And so in this case, higher values mean less iron, and lower values mean more iron. So we measured the amount of calcine fluorescence in the midgut, and upon knockdown, we can see there is a decrease in calcine fluorescence, which means an increase in cytoplasmic iron. And if you remember from the figure I presented at the start, this is an ortholog of a Drosophila gene that's involved in transporting iron out of the cytoplasm and into the ER to facilitate ferritin loading. So we think that when that process is inhibited, it increases cytoplasmic iron. The iron gets stuck in the midgut. And so that's why we see this decrease in fluorescence. At the same time, in these 24-hour ovaries, we see an increase in calcine fluorescence in the ovaries, suggesting that these ovaries are now deficient in iron, that iron isn't making it to the ovaries. And this is just showing that this knockdown was successful, highly successful in the midgut, less so in the ovaries, but still, because this phenotype was short-lived. All right, so this is at 24 hours. By the time we get to 72 hours, that iron was able to be transported out of the midgut. Ovaries had returned to normal iron levels, and when we measure fecundity and fertility, we see that there's no change. Here's the calcium levels have gone back to normal. So basically, knocking this down had delayed iron getting out of the gut, but either because our knockdown wasn't strong enough or that there's redundant systems for getting that iron out, it eventually reverted to normal, and we didn't see a strong effect on the mosquito there. I think we have done some CRISPR-Cas9 work with this gene and the other Zint genes, but the problem is for those genes is that these are developmentally also important. And so we end up, it's very hard to get a homozygous individual, and we don't have anything to report at this time. Hopefully we will in the future. Okay, so short conclusions from this part. These expression patterns we observed are consistent with a role in exporting iron to facilitate ferritin loading. But again, that's a step after we were interested. We were interested in that first step of getting iron into the midgut. ZIP11 and ZIP7 could be involved in these processes, and we're in the middle of still characterizing these. But then we started looking towards other unbiased methods, right? So we just picked these ZINT and ZIP genes because they had relatives that were involved in iron transport, but we wanted to move towards an unbiased screen to identify potential other molecules. Right, and so here's this calcine stain. This is what it looks like in cells. And so here we just rear cells on normal media, and we two different cell lines, AAG2 and A20. We treat them with calcine, we get some level of fluorescence. If we use an iron chelating agent and we pre-treat the cells with an iron chelating agent, and then we put the calcine fluorescence on, we can see as they glow much brighter because these cells are basically starved of iron because we've chelated it all up and have not allowed the cells to uptake it. And at the same time, if we treat them with a high concentration of iron in the form of ferric ammonium chloride, hard to tell from this picture, but it's easier to see in these A20 cells that the amount of fluorescence has actually gone down. So we definitely are affecting how much iron is in these cells. And so we then took these cells, harvested the RNA, and did Rna-Seq. And this analysis was done by Heather before she graduated, and Hitoshi as well. What we can see here is these are genes that either have gone up under this iron starvation condition or have gone down in expression level. And then as we move from left to right, these are weakly expressed genes and these are strongly expressed genes. So we're interested in, of these genes, which ones of these are multi-pass transmembrane proteins that might be served as transporters, right? Under the assumption that under iron overload conditions, a transporter involved in uptake might be downregulated. And under iron starvation conditions, an iron transporter involved in uptake might be upregulated. And of course, for exporters, we would expect the reverse. And so we then took out of these red dots that you see here that are below this full change cutoff, all the ones that are multi-pass TM, and then designed double-stranded RNAs to those. And we repeated our cell culture assays, except this time Hitoshi updated our 2 plasmid system with a ferritin light chain promoter driving luciferase, and the constitutive polyvictin promoter driving renilla luciferase. You put them in 1 plasmid, which increased our efficiency. And then after 48 hours, spike those with iron, and then measure luciferase activity. And so here's basically what he found. So he screened 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 genes here, and then another 1, 2, 3, 4, 5, 6, 7 genes here. Our controls are the ZIP13, which we expect to go up, and it did. And then this ZIN7, which we expect to go down, and it did, and GFP, which we expect to be our baseline. So that's what we set that as. Okay, so knowing that our positive and controls in both direction are working, we can then analyze these other genes for their effect on this ferritin induction, which again, we're taking as a proxy for the amount of cytoplasmic iron. And so a number of these genes are highly significant in terms of decreasing the amount of ferritin induction, suggesting that they were inhibiting iron uptake or increasing export in the cell. And a few of these as well. We had very few, I don't think we had any in the up direction. So this is again in cells. We took this kind of short list of genes that had the strongest effect. And Hitoshi took his double-stranded RNAs and injected those into mosquitoes. And then rather than using our luciferase reporter, which we don't have in a transgenic construct, we just measured endogenous ferritin here. Okay? And so we can see endogenous ferritin. This is the ferritin light chain gene. Again, it's strongly induced by a blood meal in the midgut. And it's not really expressed in other tissues. So we asked the question of what is the level of ferritin expression in the midgut after 24 hours of blood meal exposure. And so for 4 of these genes, we saw no change in ferritin, suggesting that the amount of iron getting in from the blood meal was about the same. But for 1 gene, this one 471, we see the amount of ferritin is about half, suggesting that less iron was getting into the gut in these cells when this gene was knocked down. So then we started to focus in on this gene a little bit. But at the same time, we also measured egg production, larval, the number of larvae per female, and overall hatch rates after each double-stranded RNA treatment. And again, here's GFP double-stranded RNA. Most of these genes did absolutely nothing, had no effect on eggs, larvae, or hatch rate. But this same gene that resulted in a decrease in ferritin expression also had severe defects in the number of eggs per female, the number of larvae, and the hatch rates. So this gene is obviously very important in a manner that was of great interest to us. So we asked the question of what this gene is. And as we lined up the amino acid sequences, we see that AA471 is part of a large family of transporters in the SLC16 family. These are, some of them have been described as monocarboxylate transporters. In flies, one of them has been described as a lactate transporter. Others transport amino acids or a thyroid stimulating or thyroid hormone. But you can see in the clade here where we find this 471 gene along with 1 gene from Anopheles gambiae and 2 genes from Drosophila, although 1 seems to be a closer ortholog. We don't quite know what the substrate is. So this is as far as the story is going now. So right now what we're doing is we're doing some metabolomics to try to look to see what small molecules might be disrupted. We're not sure if this is transporting iron directly or if this is in some way disrupting blood meal digestion as a whole. And that's resulting in just slower digestion, less iron getting into the gut through a secondary process rather than digestion proceeding normally. Because what we see, we named this gene dyspepsia. And if you read Hitoshi's paper, it just came out a few months ago. April, yeah, so two months ago now, very recent, but these mosquitoes have a lot of trouble digesting. Digestion takes much longer, and so we're not convinced that this is transporting iron, but it is involved in some aspect of digestion.
Zach,
Yes, sir.
Does any of these transports have been in any other organs, has been implicated in metal transport?
In which transport?
In metal transport.
Methyl?
Metal transport, because they are all related to transport of low molecular weight organic molecules.
Yeah. So I'm sorry, I didn't catch, you said methyl or?
Metal, iron, copper.
Oh, metal transport. No, none of them have been implicated in metal transport. Yeah.
Yes.
Yeah. So that's why we're a little bit skeptical that it might be a secondary effect, and not.
Why not think about metal complexed with an organic molecule?
It's possible.
A
Very possible.
siderophore-like mechanism.
No, it's very possible that it's siderophore bound and that it's being transported that way, and that may be the way it's being scavenged first. And definitely that's on our radar in terms of what we're looking at. But yeah, so Hitoshi is doing a number of different things. He's trying to express this in yeast and in Xenopus. We're trying to do some substrate profiling to see if we can match it to something, and then again metabolomics to see if we can see some other dysregulated substrates in the hemolymph and in the midgut as well. And so,
where is the 471 in the tree?
Oh, it's this lock down here.
Oh, okay. Great.
So interestingly, this gene is primarily expressed in the Malpighian Malpighian tubules, not in the midgut. So we think it might be involved in regulating some form of digestion or excretion, and that is creating a bottleneck that's slowing down the digestive process. But the phenotype is certainly very strong, and so we're trying to puzzle together what this transporter might be doing. I think Hitoshi has developed some antibodies to it as well, and we're trying to see which face it's, whether it's lumen facing or facing the hemolymph or both.
How does the midgut look like?
What does the midgut look like when we knock it down?
Yeah.
Yeah, so the blood stays for a long time in the guts. I think we looked at 72 hours, and they still have some blood in them. They end up excreting a lot more when we try to get them to lay eggs. So in terms of whether the integrity of the midgut is changed at all, I can't answer that. I'd have to defer that maybe to Hitoshi to kind of respond to that question. But definitely it looks like, at 72 hours, they look like, maybe 24-hour guts. They still have a lot of blood in them. The color hasn't quite changed to the kind of greenish. It's still very brown.
Have you measured uric acid?
I don't think we've measured uric acid. Yeah. Yeah, no, we definitely can look at some of those nitrogen compounds. And yeah, I had spoken with Patricia about that, Scarafia, and haven't been able to do those assays yet.
Yeah, it's simple.
Yeah.
Great.
Okay.
Okay.
So I wasn't able, I didn't go into some of the work we've done on Rna-Seq for identifying heme transporters as well. I can tell you it's just as confusing in terms of the amount of redundancy we see. What we were hoping to find was a smoking gun, something that has a single gene of a large effect size. I don't think we're in that universe anymore. I think that we've resigned ourselves to the fact that there are probably multiple genes of small to medium effect size that are mediating this process of iron uptake, and that the regulation is more complicated, but that's okay. We'll take it one step at a time. So we're moving on with, again, trying to determine the substrates of this one gene while following up on some of the other candidates that we had to see more specifically what they're doing. So that work is continuing. But I wanted to change tunes to kind of give you a look at some of the other work we're doing. This is in regards to the flight. And this started with the idea of killing mosquitoes during pupation, right? So During the larval development, we can imagine that there's intense competition as larvae are competing with each other for food and nutrients, and that by the time they reach pupal stage, they don't feed anymore, and so that competition is over. And so we reasoned that that would be a good time to kill them, so that if we kill them too early, we might make it easier on the ones that survived because there'd be more food and resources for them. Whereas if we kill them late, then those resources have already been taken up, and maybe some other larvae have died due to lack of nutrients, and then the winners here might then go on to die as well. And so we were looking for genes that we could knock out specifically in pupae, but might have no fitness cost in earlier developmental stages like embryonic development or larval development. So we mined some Rna-Seq data that had been published back in 2013 by Bruce Hayes' group. And here you can see male testes and carcass, female carcass in time after blood feeding, female ovaries in time after blood feeding. And then here's embryonic development. And then the blue indicates basically no expression, and the red indicates the strongest expression we could see. And so we focused in on genes that had very intense expression in the pupal stage with maybe minimal expression in these other stages here. And of these 6 genes, the one I'm going to talk about today is this gene called stretchin. And so what we did was we designed CRISPR-Cas9 reagents to all 6 of those genes. This is a snapshot of the microscope that we use for those who haven't done this procedure. And my apologies to Marcelo, Luciano, and people that helped develop this technology while I was still learning it. Here's a slide that shows our embryos, and here's our needle, and here's a snapshot of what that needle looks like under the microscope and what the embryos look like under the microscope. So we take a good needle, and not a blunt, and not a needle that's too big. And then rather than using a dominant marker for transgenesis, we like to do for a lot of these mutants is just identify our mutations through a high resolution melt analysis. And this is basically based on a fluorescent dye that's able to bind double-stranded DNA or a PCR amplicon. And as you apply heat and that melts, that fluorescence will go away when the strands come apart. And that based on the length and the amount of pairing, you might see a change in that melting temperature. And so here is the Aedes aegypti stretchin gene. This is a model of the different protein domains that we were able to annotate in it. Its closest ortholog is this stretchin MLCK, or myosin light chain kinase gene in Drosophila. As you can see, these genes bear very little resemblance outside of a small set of immunoglobulin-like domains in the middle here. Whereas the Aedes gene lacks the kinase domain, and it lacks a lot of these other domains. And this is size in amino acids. So these proteins are monsters, right? The Drosophila one in particular is a monster, but even the Aedes one is not terribly small. So we targeted our guide RNAs to the second exon here, right after the ATG, trying to knock out basically the entire open reading frame. All right? And so what we did was then do our Hrma assays. And this is what the output of those Hrma assays. And each curve, each line represents the leg or PCR amplicon from the leg of a single mosquito. Gray indicates our wild type. And then we have these blue curves, which we later determined contained a 35 base pair deletion. And these red curves, which we can confirm contain a 41 base pair deletion, and use those to build our stretchin knockout strains. And this work was led by Chai in my lab. He's a research scientist. Okay. And so we did this for all of these genes that we were able to make knockouts for, and then determined was there any effect on hatching, larval mortality, pupation, and pupal mortality. And what we were hoping to see was no effect on hatching, no effect on larval mortality, no effect on pupation rate, but then huge curves here for pupal mortality. So you can see how disappointed we were to see that none of these knockouts had an effect on pupal mortality. We had a few of them, despite having very low expression in the embryos, where our hatching rates were severely suppressed. So those right out as candidate genes for us. Few that had elevated mortality in larvae, but really no effect on pupation and no effect on pupal mortality. So we were kind of sad about this. We spent a very long time making these mutants and saw no clear effect, despite the fact that these genes are abundantly expressed in the pupal stage. But we kept these guys going. We didn't stop at pupal mortality. We thought, okay.
Hi, Zach. How was about the morphology, the cuticle, and host searching behavior of the mosquito? because I remember that there was an Osiris gene that was implicated in odor recognition, no?
So we did not look. We were screening these for pupil mortality. I think we still have some of those Osiris mutants. So if anybody is interested, we're happy
Because
to.
I think it was related to the cortical
Yeah, we did not see
fine
it.
morphology.
Yeah, we didn't see any defects in morphology, but we didn't look ultrastructurally. Just by looking at them under the microscope, morphologically they looked normal, but we didn't do any EM or section or anything like that. So take that caveat.
Yeah.
But in the absence of
It's
a strong
very interesting family.
But what we did see for these stretchin knockout mosquitoes is that there were a group of them that couldn't fly, right? So when we perform our cross of our delta 41 times our delta 35, you'd expect to see some that are trans-heterozygous for both mutations and many that are still heterozygous for one mutation or the other. And so we see is that some mosquitoes in those cages could fly just fine and others couldn't fly. And so because we didn't know the genotypes at the time, we then classified those into flying groups and flightless groups and then genotyped them. And we get these curves here. And the nice thing is that when you use trans-heterozygotes, you can easily see a third curve shape that's the melting profile of this product here, which of course has these, that would expect to have a bulge of 6 base pairs that gives it this unique melting profile. And what we saw was that the wild type and the heterozygous mosquitoes were the ones in the flying group, and that the ones that were trans-heterozygous were in this flightless group with this kind of altered wing morphology. And in fact, their wings, they couldn't move them at all. And so we repeated this assay, and again, on a larger scale. And what we see again is about 25% of those mosquitoes from those heterozygous times heterozygous crosses were completely flightless. And those flightless mosquitoes were almost entirely trans-heterozygous loss of function stretching mosquitoes. Whereas out of our flying mosquitoes, they were all either heterozygous or wild type, with the exception of this one male right here. And what we found out for him when we put him in a new cage was that even though we classified him as flying, he had found his way to the top of the cage where we assumed only flying mosquitoes could get to. He actually hopped his way up the side of the cage, and we put him in a new container. He was a big hopper, but he couldn't move his wings either. Right? And so this is a complete loss of function in terms of flight. And so we asked the question of, is this gene expressed strongly in the flight muscles, which is where we would expect for a gene involved in flight. And so we took a genomic fragment upstream of that stretchin gene and connected it to GFP, and then put it into a transposable element with another marker that gives us a readout for transgenesis, in this case, an eye-specific marker expressing DS-red, and then generated a number of transgenic strains. And we used actually 3 different fragments because we weren't sure how big of a fragment we would need. But the results came out the same for all 3 fragments that we used. And then here you can see under white light and under GFP and under DS-red, these different transgenic strains. And we see some variation in the intensity of fluorescence, but that fluorescence is localized in the pupil stage to an area corresponding to the developing flight muscles. You can see some of it starting in the larval stage in the L4, but the strongest fluorescence was here in the larvae. And in the adult, the body wall prevents you from seeing it, but if you take away the sheath of the body wall here, you can see that those flight muscles are still glowing bright green. And then looking at this by PR, we can see that stretchin expression is not expressed in the embryo. We couldn't detect it in L4, but it's expressed strongly in the pupae, but then weakly in the adult. I guess some residual expression is still there. So we conclude that this gene is involved in flight and expressed in the flight muscles. And this may be a good candidate for developing a strain that has this gene either knocked out or a way to attack this gene to stop mosquitoes from flying away. So that got us on the idea of just disrupting flight because we weren't quite expecting to see that right at the start. And so this idea that if, well, females can't fly, they're not nearly as frightening. And I tell this to people that work on this project, it's like mosquitoes tend to be terrifying because they could be anywhere. You look around, you don't quite see them, you kind of hear them, you know they're there, and then you just can't sleep if it's nighttime or whatnot. But if they're walking, they're not nearly as terrifying, right? So that's what we want to do, is just create a generation of walking mosquitoes that become drowning mosquitoes really, really quickly. And this is work that we published again just a few months ago with a graduate student who just finished. Okay, so the same kind of idea. We're looking at actin and myosin genes that might be involved in flight, and also a gene called flightin, cleverly named, that in Drosophila, when it's knocked out, results in a flightless phenotype. And this is just looking at the embryonic stages, ignoring the adult stages, with the assumption that if you knock something out and kill them of their pupae, it really doesn't matter what the expression is in the adult. We just don't want to hurt them in the embryo stages to allow this larval competition. We can see here is this Aedes actin 4, which was described by Tony's group actually back in the 2000s when I was in the lab there, that this was a female-specific actin gene. It's expressed to some extent in the males, but not nearly as much as in females. And that expression does start in the late larval stages. Interestingly, there's a second gene that's very strongly expressed in males and almost not at all in females. And that expression does come on in the L4 stage. And then there's another actin gene that's expressed pretty mildly, but only in pupae. And then these rest of these actin genes look like they're expressed throughout development. And so these would be more universal offenders to try to kill things maybe early on in development. Here's your primary cytoplasmic actin. And then on the myosin side, we found 3 myosin genes, one called Myosex that Jake and I had described back in 2015 that's expressed in males but not females. But there's also a gene we described in this paper we call MyoFem, because it is strongly expressed in female pupae, but not in males very much. And that expression does come on also in L4. And then one kind of generic myosin that we want to leave alone here. And then flightin, again, pupae-specific expression. So same thing, Sarah designed guide RNAs targeting multiple exons of each of these genes. Here's the gene model. These red arrows indicate the guide RNAs that she made. And then she would inject those into embryos, wait 24 hours, grind up the embryos, and do PCR and look for these difference curves, looking for guide RNAs that were highly effective. And the ones that we see these red curves that are highly different from the control embryos, we would go back, inject those guide RNAs again, and this time get germline mutants. So she was able to do this for MyoFem, for Aedes actin 4, and for flightin. This one was the hardest one because these actin genes, oh man, you can see here how many paralogs there are, and actin is pretty conserved. So she had to work to try to find guide RNAs that were specific to just this one female-specific actin. But we were able to do it. Okay, and so in the end, she ended up with a single deletion for actin 4, a single deletion for MyoFem, and 2 different deletions for flightin. And then after a number of back paralysis to kind of get rid of the genetic bottleneck, we did some tests to see what the phenotypes of these individuals would be. Okay, and this is the same type of assay where those heterozygous individuals are crossed. The surviving pupae are assisted because we assume they might be flightless. So we actually give them a little Q-tip to walk up to get out of the water, because if you don't, they'll just drown and you can't genotype them. You can't phenotype them. So after they've emerged, we wait about 24 hours, and then we decide which ones are walking on the bottom of the container and which ones were able to fly to the top of the container. This is a small image, but we actually use kind of a big bucket. So there's a huge distance for them to have to try to walk up, and it's too slippery for them to get the whole way up. They have to fly up. So we can separate out the ones that are flying from the ones that are walking, and then genotype them. And we can see here again, this is what the mosquito should look like. And then from these actin mutants, we can see again these wing postures indicating that they can't really control their wings at all. Really interesting when we look down here at the phenotypes. So again, our wild type mosquitoes, we've had nothing that was flightless. Heterozygous, they can fly just fine too. And then in the homozygous, males can fly just fine, but for actin 4, females were completely flightless. Right? So this is, we're able to get female-specific flightlessness. And then for the MyoFem, it's even more interesting because again, we had wild types couldn't fly. Homozygotes, the females were completely flightless and the males were fine. But even amongst the heterozygous, we saw about half of them we classified as flightless, suggesting that one copy of MyoFem is not sufficient for wild type flight, that they actually need both of these copies to develop these muscles. And now we did these assays about 24 hours after emergence. It's possible that that one copy would eventually result in the recovery to full flight after a few days, given a little bit more time. But it's definitely very promising because if they can't get out of the water, again, we assisted them with this Q-tip stick to get out. But if they're flightless when they first emerge, their days are numbered. And as expected, 80 flightin was flightless for both males and females with no effect on the heterozygous, right? So this is all very exciting for us. And flight, no, it's conserved. Mosquitoes, myofem, not so much. But Aedes aegypti 4, we weren't sure about. And so what Sarah did was took, again, amino acid sequences, all the acts we can find that were closely related from mosquitoes and flies. And we can see here is male specific actin here. And that this has some close orthologs in Aedes albopictus, Culex, Anopheles starlingi, and Anopheles gambi. And here's the female actin, again, an ortholog in gambi and albopictus and Culex over here. So, and when we looked and we mined the expression data of some of these anopheline genes based on all the RNA-seq and microarray data that's out there, this anopheline gene here is also female specific, and this gene here is also male specific. And this kind of split of responsibilities into controlling male flight versus controlling female flight happened a long, long time ago. And so strategies to specifically disrupt female flight targeting these genes might be applicable to a wide range of vectors. So we're really excited by that. And you can see that there's no fly orthologs anywhere in these three groups over here. The fly orthologs here in purple. These are these more conserved actin genes. You can see the branch length is basically non-existent. So this is a mosquito-specific expansion that's happened and diversification. Really interesting. We don't know what's happening out here yet, but that is pretty interesting as well. So this is one of the things that we're working on amongst other strategies because of this this disruption of heterozygotes here is we're working to try to engineer the M locus, which is of course where our male determining factor resides. And if we can engineer that with a Cas9 and a guide RNA that will be linked to maleness, and have that guide RNA or a group of guide RNAs be targeted to this myofem gene. Okay? In males, that won't have any effect at all because males don't need that to fly. When they mate with wild type females, we would expect to get males that would always contain the transgene because it's linked to M. And that they would be able to fly even with one disrupted copy of the myofem gene. But females with one disrupted copy that they inherited from their father would then not be able to fly. And so this cycle could potentially repeat for many generations to potentially get some level of control over Aedes populations. So we're working towards engineering the M locus now. And this is something we're very, very excited about going forward. Okay, so some conclusions from this last couple of projects, right? So CRISPR-Cas9, I highly recommend it if you're not already using it to study biology or physiology. It's really great for generating loss of function phenotypes. You don't need to make transgenics. So if this is something that's a concern of yours, if you're worried about doing knock-ins or just rearing transgenic mosquitoes in general, you don't need to. You can just generate gene edits, loss of function mutants, and we can track them pretty easily using HRMA. Again, we do it in 96 well plates. We do several hundred in a day, get the genotypes we want, and then run our assays on them. It's pretty simple. Stretchin and flightin are essential for flight, and they're loss of function recessive. Aedes aegypti 4 is present across blood-feeding mosquitoes. And then we're looking at these driving Y approaches potentially for mosquito population control. So one more small project. Is it okay if I go into one more project? Are we out of time? Okay, great. Okay. And so we're going to turn the stages over to talk a little bit about this parasite invasion project. This came out of a project that postdoc in my lab, Bianca Bojan, had started when she was with Margaret down in her PhD and later in her postdoc work. Looking at this gene, salivary gland specific gene 1 or SGS1. Now, back in 2006, Ken Vernick's group showed that antibodies targeted to SGS1 when they were injected into mosquitoes helped block parasite invasion of the salivary glands. And they hypothesized that SGS1, which resides on the membrane of the salivary glands, the outer membrane, and it goes into the saliva as well, serves as a receptor or a co-receptor for the parasite. So Bianca wanted to generate some either RNAi experiments or knockouts of this gene. And so what she was able to do after joining my lab was generate some CRISPR-Cas9 reagents to see if knocking this gene out actually had the same effect on parasites as incubating with those antibodies. So she was able to inject just a few hundred mosquitoes, recover a number of mutants. Again, here are these difference curves. The controls are these gray lines in the center. She was able to get 5 or 6 different groups out of this. And of those, we selected ones that were really easy to manage. So this delta 25 and a delta 23. And this is where it interrupts the protein. This is another behemoth of a protein. It's a single exon. The gene is thought to be a horizontal transfer event an ancient horizontal transfer event from bacteria to an ancestor of the mosquitoes. And so it's encoded in one long exon. I think the protein is, yeah, you can see here 3,000 amino acids long, so it's not a small gene. But the edit happens about here. So then, in collaboration with Eric Calvo's group, he was able to take our salivary glands. We wanted to make sure the protein was gone because we didn't disrupt it right at the start. And he was able to do proteomic analysis on the salivary gland extracts from either SGS knockout mosquitoes or from our wild type, and just basically look at not only is SGS1 gone, but have we disrupted any other aspects of the salivary glands that might confound our interpretation of our resulting experiments. So we can see is every other gene, we can see this nice one-to-one relationship between the number of peptides we find in the wild type and the number of peptides we find in the SGS1 knockout. We can just draw a nice line right here, with the exception of SGS1, where we find almost 450 peptides in the wild type and less than 20 in the knockout. And when we map those across the length of the protein, what we can see is that in wild type, we see peptides up to about 3,000. We don't get the last 300 amino acids for reasons that are still fascinating for us. But in the knockout, we can see we still get peptides from this area before the CRISPR-Cas9 knockout, but then because of the frame shifts that occur right after the knockout, we don't get any peptides afterwards. Okay, so we're convinced that this protein is basically gone and would not be made in a form that would be able to help the parasite. What then Bianca did was then travel to NIH, to Eric's lab, and perform Plasmodium infection challenges on these mosquitoes. And here you can see 3 different experiments she performed along with that group. And so these were all, each group was fed on a different chicken that had a different parasitemia. And here we can measure oocysts in the midgut. And then here we're measuring sporozoites in salivary glands at day 8. So here's oocysts at day 6 and salivary gland sporozoites at day 8. And this is Plasmodium gallinaceum because we're working in Aedes aegypti here. And so what we expected to see is what we see over here on the right. We see reduced sporozoite numbers in all of the knockout experiments. Okay? You can see the parasitemia down here in the bottom. So the strongest parasitemias, we have a mild knockdown. The strongest knockdown was a lower parasitemia because the challenge was less. So this is what we expected to see of a molecule that we thought might be involved as a receptor in the salivary glands. But we were a little bit surprised to see what we see over here on the left. And again, this is 3 independent experiments here, so we're very convinced this is actually happening. But we see a strong reduction in the number of oocysts in the midgut. And this was surprising to us because SGS1 is pretty much exclusively expressed in the salivary glands. It's not expressed in the midgut at all from what we can detect. And so what we imagine is that as some part of the SGS molecule is actually secreted in the saliva, that must be having some effect either on the bite site, which changes parasite infectivity, or the mosquito is re-ingesting SGS1 protein in the midgut lumen, and it's having some effect on parasite infection there. And we are very excited to be following up on trying to explain why we see these reductions in oocyst numbers when we knock down this salivary gland. We know that mosquitoes do re-ingest all their salivary gland proteins. We've done proteomics on the peritrophic matrix after artificial meals. We can find them there. Other people have found salivary proteins there, so it could be from re-ingestion, but we're not sure what's going on there. But it's an exciting new mystery for us. And I wanted to mention some of Bianca's work here because again, it was done in collaboration with Margaret, and it's some exciting work that we're moving towards now, a new direction. Again, this paper just came out a month or 2 ago as well. So tried to highlight some of the recent projects that we've got going on. They're all ongoing. Anytime I give a presentation, I don't like to end with a story that's over because none of my stories really end. They just kind of keep going forever. And so I just wanted to highlight some of those projects. So in the end, again, to review, so initial steps of iron acquisition during blood meal are still uncertain. We have several candidate genes identified through either targeted or unbiased screening approaches. And we're still working towards characterizing those. And we, of course, publish that data, and we hope that others will join in the search. We've identified at least 1 gene, which we call dyspepsia, that's involved in somehow in iron metabolism. I don't know if it's involved directly in iron transport, but it's involved somehow in iron metabolism that appears to be critical for female fertility. We use CRISPR-Cas9 routinely for analyzing the function of various genes, and we've identified several important in flight. And then this gene, SGS1, plays an added role in parasite development in the meta. So with that, I want to acknowledge Poon, who did the stretching work, Toshi, who did iron work that I discussed, Bianca did the SGS work, and Sarah, who did the flightin and actin and myosin work in the lab, as well as Margaret, when she hosted Bianca, and Eric for hosting for Bianca for a lot of that work as well. Of course, all of our other collaborators, without this, would not have been possible. And then funding sources from different federal agencies. And of course, all of you for your wonderful attention over this very long seminar. So thank you, and I love for more discussion.
Questions?
May I make a question?
Sure.
Okay,
Please.
well, thanks, Zach, for the amazing talk, beautiful work, congratulations. And I was curious about this genes involved in female flight, and I was wondering if you have made some kind of behavioral studies and differential expression of these genes related to, I don't know, how can I say, different situations in during the female life cycle, the adult.
This is something that we're very interested in and that we're moving towards now. Like I said, all this data is basically fresh off, so this is up to date with where we are. But we're very interested in whether or not these genes are involved in modulating flight tones, for example, during courtship, whether they're involved in mate recognition. We don't have those answers yet, but we're very interested in those experiments.
Okay.
Zach, could you close your presentation? Yeah, I think is better to discussion. Sorry.
Oh, yes.
Okay, thank you.
Please.
Yeah.
Yes. So I was wondering if you did any blood feeding assays or behavioral, because I saw that flightin and actin-4 were differentially expressed during the after female emergence, and they were specifically expressed in females but not in males, and also that they were expressed in the female head but not in the body. So I was very curious when you showed
So for those mosquitoes that are flightless, doing a behavioral assay is very difficult because they can't exhibit a lot of normal behaviors. They're stuck at the bottom of the container. They can't really host seek. I guess we can probably create some specialized arenas where they can walk to somewhere where they could potentially feed. We haven't tried to do that yet, to try to manage them because they are walkers and not flyers, and a lot of interesting behaviors happen in flight. What we're looking at is largely to have them die, and if we didn't help them out of the water, none of them would get out of the water. I mean, they would just die upon emergence. And so I'm not sure if we can say that these genes play a role in certain behaviors, because biologically speaking, without those genes, they would just drown.
Thank you.
Hello, can you hear me?
Yes.
Really nice speech. I just have a question. It's curious actually, because you mentioned in the first section of your presentation about the eggs laid. So you had the reduction in the number of eggs laid, but my question is, did you see if the number of eggs changed or just the number of eggs laid? Like, did you open the ovaries to see if any eggs were inside the ovaries or just the number of eggs laid?
That is a good question. I will have to go back and ask Hitoshi that. I think that they did not develop. I'm not sure if he dissected ovaries or not, but because we think it's tied to the fact that they hadn't even finished digesting the blood yet, right? And so, when we're doing these assays, we're doing them at 72, 96 hours after blood feeding. And for these groups that we had the knockdowns, you can still see, mean, they look like they're at 18, 24 hours after blood feed. They just have not got very far. And I think that the oogenesis is kind of a process that they haven't gotten the nutrients to initiate that. But I can't say for sure because I can't recall if he did the ovary dissection or not.
Marga?
Is a comment. You have a flyless mosquitoes.
Mm.
You cannot just try to see how much is the bother their behavior. Is a flyless mosquito.
That's right.
You know, that's it.
Yeah, that's
This
it.
is a comment. That's it. If it's less eggs, they are not going to produce anything. They are made to be killed.
I remember that one of the first studies that Alexandre Peixoto made with circadian rhythms in mosquitoes, he cut the wings to put them in the infected and not infected, to put them in the activity tunnel. So that Rafaela, that's there, I think. So you worked in that experiment, no, Rafa?
Yes, but in that experiment, we were looking for locomotor activity, which is in fact a little different from flight activity.
Yes, no.
In that,
to comment on Margaret thing that
yes,
you cannot measure flight, but locomotor activity in that assay you used it. It's possible.
yes.
Yes, I was wondering if you can compare the expression of different genes between the flyless mosquitoes and the mosquitoes that can fly but without wings. So we could see like difference between these mosquitoes and more genes that are important for the flight behavior.
Yeah, we haven't done that, but certainly you could do that. Yeah, if you wanted to get an insight into how that gene influenced the expression of other genes, if there's any other collateral effects.
Yeah,
But
Yeah, Fernando Gento stole my question. I was about to ask about the behavior assays, but Fernando asked first, so.
Sorry, Rafa.
Okay, okay.
just may I ask you about the Osiris mutants?
Yep, sure.
Because I had a student that made some Osiris work with Rodinus.
Okay, okay.
And so the work was unfinished. The student gave up and left the lab. Mm-hmm. But it was I think it's a fascinating family of proteins. Mm-hmm. So did you see any phenotype on that knockout?
You know, there's probably assays that we could run that we might see something. We were looking for you know, mortality, and we were looking morphologically to see, you know, obvious things like cuticular defects. We expected them to be involved in some aspect of that. So, but morphologically, they looked normal. Like I said, we didn't look ultra-structurally, and we didn't look at any types of behavioral type assays. So it's one of those things where we were screening, you know, 6 different groups, and we followed up on the group that gave us the clearest phenotype, which was the stretching. But I think we still have those, and so if somebody has a good suggestion of something we could run, definitely we could take that into consideration.
Okay. Yeah, the original name of the family was described as a triple lethal. Mm-hmm. You know, so yeah, exactly the same problem. So
right, that's right, redundancy. So we would think that single mutants are not going to do it; that we would have to knock out a certain number of them because of the redundancy.
Okay. And do you have any idea what that because maybe I lost something that salivary gland protein, what does it do? What is the role of that protein? for the mosquito?
That's a great question. I can't tell you the role. We don't know the role of it right now. Because it's such a monster, you know, Eric has been trying to express the pure protein on its own and run some physiological assays on it. It's just too big right now. We haven't been able to make it. And so we're still working on that. And, you know, we have the mutants, we know what it doesn't do because we've run some assays and we see there's no effect on X, Y, and Z, but what it does and why it's there and why it's been preserved in the salivary glands. And it's one of the most abundant proteins we can find in the salivary glands. Yeah, hopefully we'll have a good answer for you soon, but we don't know right now.
Okay. Yeah, interesting. So, and so if I understood so just a curiosity, why the name dyspepsia?
Oh, so dyspepsia is kind of a synonym for indigestion, and we gave it to because the mosquitoes have so much trouble digesting blood.
Okay. So everything in digestion. So have you measured trypsin activity or something like that?
We haven't measured trypsin activity, but we just can tell, you know, based on, you know, that's what the gut looks like and the color of the gut contents and the stage that it's at. Everything is just kind of delayed.
Yeah. So the peritrophic matrix also does not form.
No, I think it forms. I think it forms.
Yeah. Oh, okay. Okay. Yeah. You know, back again about the uric acid transporter of mosquitoes has been identified. I don't remember that. I remember one ABC candidate in Bombyx.
Yeah, I don't know. I don't think so. I think, you know, I'm familiar with Patricia's work. That's mostly on the enzymes involved in the synthesis and breakdown, but I don't think I've seen anything on transporters.
Yeah, but in humans, I think no? Remember the superfamily, maybe not the 16, maybe a different one.
Yeah.
I was thinking because we just thought that we published some data in Hodgins, where we speculated that maybe the production of uric acid works as a pacemaker for blood digestion.
Oh, very interesting. Okay.
So something like that.
Yeah.
And then if you change the concentration, the metabolism, because it's both an antioxidant,
yeah,
and a nitrogen excretion product, meaning that if you cannot get rid of that, and Malpighian tubules are the ones who made it, then it's something that stopped it. So and that was what we made. It's a work that we had some evidence for that, but it was also a work that was stopped half of the way. So it was not complete. We could publish it, but it was part of a PhD thesis, and the student left, went to US, and it's now working with Drosophila and another thing. But so we could publish, but I think it was, I'm not completely sure of the conclusions we put there in the paper. So.
No, definitely good suggestions. Definitely good suggestions.
Yeah. Another candidate of an important organic molecule that would be interesting is Centurionic acid.
Yeah.
Because it's produced intracellular, it binds iron, and it's released during digestion. So if you blocked its transporter, then you would, if it is a way to excrete iron, that I never thought about that's occurred to me during your talk.
Yeah, I'm not sure about that one because Aedes aegypti, you know, the white-eyed strains that we use are deficient in XA.
Yes, we looked for the heme, of course, because we have that kind of fixation with him.
Yeah.
But at the same paper, we showed that it binds iron, and that's it's a beautiful thing. It gets green, so it's very easy. So, but it's produced intracellular for sure.
Yeah.
Because the enzyme is intracellular, and then there must be a transporter.
Right.
Of course, nobody looked into that because the Centurionic acid is found in the lumen of the gut. So we thought we discussed on the original paper several years ago that the idea would be it would be a chelator for iron in the gut.
Yeah.
But it may be the other way around. It may be a way to get iron out of the gut.
That's right. That's right.
So, and then it could be a CDR4. A very similar molecule called quinolobactin is used by bacteria as a CDR4 to transport iron. A very, very close molecule.
Yeah, no, this is great. This is great. We'll definitely look at that too. But what do you think about because the mosquitoes that are white-eyed? I don't know because they're deficient in XA, but we would expect to see some of these same defects if that was the case, right?
Some of can you repeat, please?
The white-eyed strain we have it's deficient in kynurenine hydroxylase. They should also be deficient in XA.
Deficiency in
xanthuric acid.
Oh yes, yes.
Those blood feed normally.
Yeah. Yeah, but then you can measure the amount of centurionic acid in the lumen and in the tissue.
Yeah.
In your mutant. Don't need to make anything else. So just go there. It's very simple. And if you do an HPLC, it's amazing because it's by far the largest peak.
I see.
Only with optical detection. So it's close to sigma purified centurionic acid. You do if you precipitate with acid, take the supernatant and apply to a C18 column. It's simple.
Okay, that's the gut content, or
yes, yeah. We made that with we took the gut content or whatever the gut. I don't remember exactly if it was gut content or the whole gut. I think it was in the gut content, and at the end it appears in the feces of the insect.
Right.
So it's truly excreted. But for sure it should be found in the tissue somewhere. We never looked.
Okay. Yeah. That'll be interesting because we can add that because we've been doing hemolymph profiling, looking for disturbances there, but we can definitely look in the gut content too from the protein-free meals.
Yeah, hemolymph we never looked, but it can be produced in other place and transported through maybe the gut or maybe the Malpighian tubes.
Yeah.
Because it's
Genta. Zach, let me go back to the female flight. I'm having thoughts about that. Have you looked for another vector species like sandflies? If there is any difference between sexes?
Oh no, we didn't back up to sandflies. We looked at Drosophila, but we stopped at mosquitoes. So that's wide open. I don't know if it's that way in sandflies or not. So it definitely would be interesting to look.
Okay.
Yeah.
I think there's a comment in the chat. Rafaela, you were interested in a copy of the paper that we published. If you want to just send me a quick email, or I can send it through Gabrielle and she can forward it to you.
Okay.
Either is fine, but I'm happy to provide that.
Thank you.
I have it. I can send her.
Okay.
Any more questions? Pedro, you can finish the seminar. Okay, no problem. No more questions.
Okay, well, thank you, everybody.
Thank
Really
you.
enjoyed talking. I really wish we could do this.
Very
in
good presentation.
Thank you very much.
Thank you.
So great.
Okay, thank you.
Nice to meet
Bye-bye.
you. And
Bye.
keep in
Thank
touch.
you a lot.
Bye.
Bye-bye.
Bye.